Okay, so today's title, or the title of today's lesson is DNA Replication. Today's February 25th, 2021, and it is the second day of our fifth unit. The objective today is to explain the double-stranded complementary nature of DNA as related to the process of DNA replication. And then the essential question, how are copies of DNA made? Yes, there's one right behind you. You see it? So we briefly talked about DNA replication in Unit 3, which was all about cellular division, the cell cycle. But here we are introducing it again. We're going to go into greater detail this time too. I see some folks are still writing here, so giving them time, and hopefully you at home time as well. I like that jersey, JT. PSG. Are you good? Abina, are you good, or you need a little more time? Okay. All right, so I have a question for you to begin the lesson. Why does DNA need to be replicated? What's the point? I kind of already went over it, but what do you all think? Why do we need to replicate our DNA? The answer is basically right behind me, so those of you at home probably won't be able to see it, but those of you who are here might be able to, based on your angle, see it. Good, thank you, Janelle. Yeah, there's got to be a copy of DNA of the new cells, but I do like the way that Janelle put it, using that word chromosomes. We have to maintain the number of chromosomes. So if a cell has 46, we want the daughter cells to have 46 chromosomes as well at the end of mitosis. In order to achieve that, we can't just split in half. We've got to first double, then we split in half. So we go from 46 to 90, and then we can split again in 46. And I would just remind you that I do recommend that you try to write down everything that's highlighted at the very least. Can't tell what folks are. Okay, I'm gonna go ahead. All right, so DNA replication is a very complicated process, and it involves a lot of moving parts, even though this is happening constantly throughout our bodies. Our bodies are made up of about 3 trillion cells, and this means that cells are always dying and needing to be replaced. So this process of DNA replication has to happen before any new cells can be created. So it's happening all the time, but it is still a very complicated process. We're not going to actually talk about each of these moving parts. We'll talk about a couple of them. But I want you all to notice a couple of things. Number one, on the right side of the screen, we can see old DNA. We see that it is still wound up in a double helix structure. Then we see that it gets unwound, then it gets unzipped. And on the left side of the screen, we can see new DNA being formed. 2 new molecules of DNA are being formed. Okay, so the other thing I want you to notice is the number of words that end in ASE. What do all of the ASE endings mean? Does anybody remember?
Enzymes like proteins or?
Good, Josue, thank you. Yeah, so when we see words that end in ASE, they refer to enzymes. And as you said, enzymes are a type of protein. Good. So we've got a lot of enzymes that are involved in this process. DNA polymerase, DNA ligase, DNA primase, helicase, topoisomerase. There's another one called exonuclease that's not in this picture. But there's just a lot of enzymes. And of course, those enzymes are needed to speed the process up. All right. And one more question, where does DNA replication occur? So we now know why it occurs. Janelle told us that we have to maintain the number of chromosomes, but where does this process happen? What do we think? Excellent, thank you, Abigail. So in the nucleus of a specific type of cell, what type of cell would that be?
Eukaryotic.
Good, so in eukaryotic cells, that's where we see this process happening in a nucleus. But there's another type of cell that does not have a nucleus. So where do we think DNA replication happens there? In the cytoplasm. Good. All right. So wherever you find DNA in the cell is where you will see DNA replication taking place. In prokaryotic organisms, this has to happen in the cytoplasm. Prokaryotic organisms do not have nuclei. In eukaryotic organisms, this has to happen in the nucleus. All right, so here's a little joke for you. It's probably not a funny joke, but it's a joke nonetheless. So a prokaryotic cell says, you tried to copy my DNA, but you did it backwards. And the eukaryotic cell says, and? You get it? DNA back. DNA backwards is and. Okay, all right. Thank you. Just a couple laughs for you there. Okay, so we refer to this process of DNA replication as semi-conservative. This means that half of the original parent cell's DNA is saved, it's conserved, and is used in the new daughter cell's DNA. This means that the newly synthesized DNA molecules will be half old DNA and half new DNA. Okay. So there are really 3 or 4 basic steps of DNA replication. Obviously, like I said, it's a complicated process. We could go into several other steps, but for the purposes of this class, we are going to focus on the basics. So the first part, obviously, DNA starts off as a double helix. And so therefore, it needs to be unwound before you can do anything to it. It needs to be unzipped. The enzyme that comes through and actually unzips it is called helicase. We see that ASE ending, indicating that this is an enzyme. Helicase, as we can see in this animation on the screen, comes through and breaks the hydrogen bonds that hold nitrogen bases together. And this separates the 2 strands of DNA. Okay, cool. The second step involves an additional enzyme. This one is called DNA polymerase. As we will see, there are 2 types of DNA polymerase, but we won't really specify the types. in this class. The first one that comes up is there to add new DNA. So it's going to add complementary base pairs to both of the newly exposed strands. So after helicase comes through and it unzips the DNA, we end up with 2 exposed strands. And now DNA polymerase comes through and it adds DNA to those exposed strands. DNA polymerase is able to add the correct bases because we know that A complements T and G complements C and vice versa. So anywhere there's a C, DNA polymerase will add a G. Anywhere there's a T, DNA polymerase will add an A. Cool. You need more time, JT? You good? I'll just wait. It's funny. Okay. Then we've got another enzyme. Whole lot of enzymes involved here. This one is called ligase. Ligase's job is to come through and glue the pieces together. And so it's going to help the new DNA kind of re-zip itself. This time, of course, we've got 2 DNA molecules. We unzipped 1 DNA molecule. In doing so, we allowed DNA polymerase to come in and add new nitrogen bases. So now we have 2 DNA molecules. Okay. And then lastly, this time there's another DNA polymerase. They are different. One is called DNA polymerase 1 and the other is called DNA polymerase 3. But we don't need to know that. We just need to know that in this step, DNA polymerase comes through and proofreads the strands to make sure that there are no mistakes. Those mistakes lead to mutations, and we'll talk about that next week. This process is remarkably accurate. Like I said yesterday, we've got about 3 billion base pairs in our DNA. and very, very few mistakes occur. Very, very few. All right. Okay. So this is the process, represented very simply. DNA is essentially like a ladder. We talked yesterday about the actual structure of that ladder. There is a sugar and phosphate backbone, and then the nitrogen bases are basically the rungs of the ladder. So the first thing we do is we break those nitrogen bases apart. We separate them using what's called helicase. Then we come through with another enzyme called DNA polymerase, and we add new bases. We add new rungs to each side of the ladder. And what you end up with, because this is a semi-conservative process, is two DNA molecules, both of which have half of the original DNA molecule and half new DNA. Okay, let's watch this video.
DNA is a molecule made up of two strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The two strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand, and wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The two strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called Helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called Polymerase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously, the DNA polymerase adding bases one by one in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA Ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
Let's practice with some of this base pairing now. So keep in mind that A complements T, C complements G, and vice versa. So I've got a parent strand of DNA, TA, CCG, AAAT, CGCA. Which should be the first base that I put there in the complementary strand? A, T, G, G, C. Oops. A, good. All right, so it's pretty simple, right? So should I bore you with the next one? This one's a little bit longer, but it's the same concept. Someone can just rattle them off, and I'll just hit the button. Someone, anyone? Deasia, thank you. Do you understand this concept? Yeah. Okay. So what should be the first base pair? A. Good. 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 Wonderful. And thank you as well, Josue and Dejan. You guys looks perfect. All right, so this is simple. Now, one cool thing to mention before I move on, the first four bases in this strand, TATA, we call that the Tata box. It's a funny name, I know, but it's an important area of most genes in our body. This is typically kind of the symbol that a new gene is starting. And so we see this same Tata box appearing at the beginning of many important genes in our bodies. Keep that in mind. All right, so I'm gonna push you guys a little bit here. We won't talk about this process until, I guess we do talk about it tomorrow. But we know that RNA is a slightly different molecule than DNA. RNA has a different sugar. It has ribose sugar. DNA has deoxyribose. RNA has the nitrogen base U, uracil, instead of T. So in this case, when we're going from DNA to RNA, what we need to do is put U's instead of T. So what should be the first base that I put in this RNA if I'm transcribing AT, GC, blah, blah, blah? What should be the first one that I use? U, good. Thank you, Ashanti. And then what? An A, yeah, that doesn't change. The T still goes to an A. Then what? U, A, C, G, Good. Thank you, JT. So, and thank you as well, Ashanti. So what we can see here is that there are no T's in RNA, only U's. We'll talk more about this tomorrow, but this is the process of transcription to go from DNA to RNA. All right, so that was a short lesson. We still have 25 minutes left. I want to do something really quickly. We do have an exit ticket. I didn't assign any asynchronous assignments, so I want you guys to use whatever remaining time we have to work on missing assignments. But before we do that, I have an idea. We have this kind of cool setup now. We've got some people who are in person and some people who are at home. So the in-person people can still see the whiteboard even if I turn my camera off. So what I want to do, I'm going to write some terms on the whiteboard and I'm going to ask them to describe the terms to you all. Obviously, you won't be able to actually see it, but they're going to have to describe it to you in such a way that you'll be able to know what term they're talking about. So for example, and this is like, it's, what's this game called? Not charades, but does anybody know? I can't remember. You can't say, they won't be able to say the actual term. They can only just describe it. They won't be able to actually say the words. For example, let's say I wrote enzyme on the board. They could say ends in ASE. And then you all would have to tell them that it must, is it called heads up? It might be. Oh, the phone version? Yeah. Yeah, it basically is the same thing. So let's see if this works. I'm going to write some terms on the board, and then I'm going to ask one of them to, would any of you like to do? You just don't want to do it. JT's going to do it. Okay, JT will do it. All right, I'm going to turn my camera off and write some stuff on the board. One more. Are these too hard for you? You think you can do these? Do you remember what it is? Do any of you remember what this is? Okay, good. Adina knows. All right. One more. All right. Come up here. I guess I didn't need to do that. I can just turn it around like this. Let's see how long it takes to get all these songs. You can come up here. You're gonna have to because the microphone is not very good. All right, so JT is getting ready to. I guess they can't see if I face it this way. So JT is getting ready to describe these terms, And I want to see how long it takes you guys to get them all. All right.
No.
No. Well, you don't have to go over. You don't have to go over. Yeah, you can jump around if you want to.
All right. This is the type of ends and O's.
Put it in the chat.
Cool. I
Okay.
can see it. I can see it. So.
That's sweet.
Okay, they're showing me here. You might have to do something really loud.
Oh. So this is the type of sugar.
This is a type of sugar. Okay, they got that one.
All right.
This is way too slow. It's supposed to be rapid fire.
Next one is a monomer of protein.
It's already been 3 minutes. Okay, I got that one? Yeah.
All right. This gives function. This is going to be ribosomes.
This gives energy to ribosomes. They might be another clue. What's something else you could say about that?
The powerhouse of the cell.
Okay.
All right.
So,
To be afraid of water.
Did you figure it out, Abina?
Organism that cannot produce its own food.
You don't need to say it negatively.
An organism that can produce its own food.
Okay, is there another thing we can think of? It's the opposite of say it louder, Abina. Yeah. Opposite of
I got it.
you guys are going so slowly. Okay, but that's how many? 5. There are 3 more.
Well,
All right. What should I make it? What else have we gone over? Cellular respiration to the body.
Mhm.
Cellular to the human body. There's something else we could say for that. You guys aren't close. Please help. Okay. To do what? Okay, so Abina is adding uses sunlight to make food. They should definitely get it based on that. Thank you.
Yeah.
All right. So that's 6, 2 more. We're like 10 minutes in. Yeah, 9 minutes. What about for the other 2?
So,
Which one are you talking about? Okay. What does it produce? What kind of cells?
this is DNA.
Good. So you said the process that produces gametes.
Got it.
Okay, good job. Last one. It's funny because we just talked about this.
Maintain the water.
I don't know, they might not have heard it. To maintain the right amount of chromosomes in the parent cells. Good. I'm going to write it up here, rotation A. Rotation A, second. No, this is third one, right? Okay, so here are the terms. That was not good. We need to improve the time. I'm going to see what how well my second block does. Rotation A3. 11 minutes and 1 second. Okay, that took 11 minutes and 1 second. So tomorrow we need to improve upon that. But now that you guys know how to play, you see what we're going to do. We've got 9 minutes left. Right, 9 minutes. Yeah, 9 minutes. So why don't you all just use this time to do the exit ticket if you haven't already. Work on some missing assignments. There will be an asynchronous assignment tomorrow. So I want you guys to be smart about how you're using your time so that you don't fall any further behind. And if you guys who are actually here need any help, just let me know.
Good, man.
All right, peace out, folks. Have a good day. I'll talk to you all tomorrow. It's Friday. I know we're all grateful for that. See you, Deasia and Ashanti. See you, Bina. Have a good day.